Hi, my name is Grant Pearse. I'm a remote sensing scientist at Scion Research. Thanks for inviting me to talk at the 2020 virtual edition of the FGR conference. My presentation covers FGR-funded research on nursery automation and phenotyping. We wanted to develop methods that would allow us to rapidly assess and count nursery crops in a way that was suitable for both automation and phenotyping. And at the same time, we wanted to test new sensors that had become available, which could allow us to count seedlings and measure their height at the same time. This project expanded core-funded research that we did in 2017. In the previous research, we showed that cross-correlation template matching, a very simple image processing technique, could achieve 90% accuracy where the image quality was high enough. However, this method achieved low uptake because it required dedicated software and it was very slow to make predictions. During this same work, we also noted that when you generated orthomosaics from your overlapping UAV imagery, you could actually get a reasonable estimation of seedling height within the beds. So based on this work, we know that getting counts of our seedlings from UAV imagery is straightforward. And we also knew that we could get some idea of seedling height as well. But there is one fundamental issue that we identified when we were doing this work. And that's that all of these results are based on a process called orthorectification. And there are significant drawbacks when trying to use orthorectified imagery for phenotyping or automation. Orthoimagery offers us consistent scale and perspective, but it also requires that we fly our UAV up and down the seedling beds with up to 80 to 90% overlap. In addition, we also have to take this imagery and run it through dedicated processing software that can take hours or days to produce the full orthomosaic. In addition to taking a lot of time to capture, orthorectified imagery also loses an enormous amount of detail and spectral purity. If you contrast the quality of the imagery obtained here on the left-hand side, this is orthorectified imagery captured from Scion's UAV, with the detail that we get off the raw imagery straight off the UAV here on the right-hand side, we can see that we lose detail that would allow us to detect this weed outlined in red. In addition, we can't use orthorectified imagery to extract these reflectance spectra here in panel C, which would allow us to detect nutrient differences at the level of the individual seedlings. To test our objectives, we established a trial in Scion's Nursery containing 3 beds of radiata seedlings. Within each bed, we selected a minimum of 30 rows of seedlings, and each row of seedlings was measured at monthly intervals alongside the ground truth data captures. We tested 3 camera-based systems for measuring seedling height. The first was the Intel RealSense, a camera based on stereo depth estimation. The second was the Stereolab Z, a stereo camera based on visual inertial SLAM, and the last was the Microsoft Azure Connect, a camera based on time of flight ranging, which is a class of sensors that's increasingly available in consumer-grade smartphones. After a successful trial setup in November, we carried out a calibration run in December 2019. We initially scheduled monthly field and UAV height measurements. However, due to the level 3 and level 4 coronavirus restrictions, we lost 3 of the measurement dates. Because of this, we deemed that it was impossible to develop an accurate height growth curve for the seedlings, and we could not conclusively test the sensors. We had to abandon the height estimation project and refocus on other aspects. So we knew from our previous research that counting seedlings off orthomosaics is relatively straightforward, but we also knew that we could improve in certain areas. So we wanted to develop methods that would make it easy to deploy, that would be scalable and generalizable to large seedling beds, and that could handle complex scenes, potentially containing weeds. We also wanted to get methods that could go beyond counting to allow us to actually delineate individual seedlings, which is a key requirement for phenotyping. Before we can count the seedlings, we have to tackle a much more fundamental task, which is how do we acquire imagery over these very long seedling beds that can contain tens of thousands of seedlings. The first method we trialed uses direct georeferencing. In this approach, we take our UAV and camera and we equip it with a high-precision GPS. Then what this allows us to do is fly the seedling bed in a single pass with overlap requirements reduced from 80 to 90% down to 40%. In addition, the imagery that this process produces is georeferenced to within a few centimeters of its true location. Direct georeferencing greatly reduces the impact from the orthorectification process on the final image quality. It means that we don't have to do any aerial triangulation, but we do have to have a high-precision GPS, and in addition, we also have to have a carefully calibrated camera. Requiring a high-precision GPS and a calibrated camera does impose some limitations on widespread adoption. So what we did was we trialed a second method based purely on computer vision principles. Here what we do is we fly the bed once with 40% overlap at an altitude of 2.5 to 3.5 meters, and then we apply some corrections to get undistorted images. We take this undistorted imagery and we use a combination of algorithms called sift and ransack to build a continuous panorama of the seedling beds. You've used similar algorithms if you've ever generated a panorama using your smartphone. These algorithms look for common features and overlapping imagery, and they use these to essentially tie the images together into a continuous panorama. Once we had methods to capture imagery of these long seedling beds, we needed to develop algorithms that could count the seedlings they contained. Our first approach focused only on speed and accuracy, and we chose a neural network architecture called the darknet. This is a very fast framework that allows us to rapidly count seedlings off of the panorama. The second approach was slightly more sophisticated. Here we wanted to move beyond detection to do instance segmentation, where we actually draw an outline around a circular area containing the bulk of the needles for phenotyping applications. To do this, we had to capture labor-intensive annotations by drawing polygons around the outlines of hundreds of seedlings. Then we fed these to Detectron-2, a neural network system released by Facebook AI's research. The last topic we addressed is something called microplot extraction. Here, what we do is we aim to capture the best of both worlds. We generate an orthomosaic from overlapping UAV imagery, and then what microplot extraction lets us do is it take the real-world corner coordinates for each of these blocks that might be allocated to randomized treatments, and then we use a series of software scripts, and we go from XYZ coordinates in the real world to 2D coordinates in the raw input imagery. And this lets us automatically extract raw, unrectified imagery for use in phenotyping. The results from the direct georeferencing approach to generating long imagery over the seedling beds were excellent. Overall, it produced very high quality orthomosaics, minimal artifacts, and it was georegistered to the centimeter level. Most importantly, it allowed us to do this with up to 50% less overlap in flight time than a traditional orthorectification approach. We also used the imagery from direct georeferencing to test microplot extraction. What you can see here is a orthomosaic generated from the direct georeferenced imagery. And here we've got a simulated trial block. What we can do is we can take the corner coordinates here. We can use microplot extraction to go from the orthomosaic to the raw unprocessed imagery. And then we can overlay the 3D coordinates of the trial block into 2D positional space here on top of the raw imagery and extract just the pixels that belong to this treatment block. What about our second approach, where we try to take a single pass UAV imagery with moderate overlap and we try to turn it into a panorama? Well, the first approach, where we used sift and ransack from OpenCV, worked fairly well. But what we found was that the no-code approach that we trialed using the commercial software PT-GUI produced extremely high-quality panoramas with virtually no loss of detail and very few seam lines. This is important because it's a no-code solution with a relatively low cost, and it produced excellent results in a short amount of time. A common issue with both of the methods that we trialed was the generation of automatic type points over seedling beds. What you can see in this figure here are the automatic type points generated, in this case from Pix4D, but the results were the same for all of the other methods that we trialed. You can see that most of the matches occur in the ground around the seedling beds. And this figure over here shows that where the software thinks it's found some matches, it's actually looking at very different objects. But those matches found around the ground are largely correct. We actually ended up leveraging this by laying down regular markers alongside the beds to facilitate improved image matching using all of the techniques that we trialed. We couldn't wait for the end of the trial to do all of the development work, so what we did was we arranged to do a mid-rotation capture. This was done in November, and here what we did was we actually asked the nursery staff to hold off applying herbicide so that we could let the weed level build up in between the seedling rows here. Then what we did was we annotated just 25 images and fed them to our instance segmentation model. We took these 25 annotated images and we used them to train our mask RCNN instance segmentation algorithm. We were very surprised to see that we got 86% detection accuracy on a test set of images that were completely withheld from model training. This turned out to be an important finding because it showed that these counting algorithms can also be trained to work with much younger crops. The imagery generated from the panorama and direct georeferencing approaches was far too large to be used with our deep learning models. So what we had to do was we had to use an approach where we take these long images and we tile them before we run them through our models. What we do is we take a subtile, we record its position in the overall panorama or long orthomosaic. Then we take that subtile and it's small enough that we can run it through our trained model. The predictions we get back can be recombined with the information on the tile's original position to transfer the location of these individual seedlings back to their source within the overall panorama. The darknet model that we trained just to rapidly detect seedlings produced excellent results. The precision of the model was 96% and the sensitivity was 92%. Importantly, we achieved this using a Tesla K80, which is the lowest cost cloud GPU for training these types of models. So although it took 6 hours of training time for the model, we were able to detect all of the seedlings in a bed in around 2 to 5 minutes. This is important because it means that not only is the darknet algorithm accurate, but it's scalable, potentially to tens of thousands of seedlings. This slide shows the results of the darknet algorithm applied to the full panorama using the tile predict aggregate method that we described earlier. What we can see is that there are some false negatives towards the edge of the beds, particularly in seedlings that were too short to be topped. This is because our training data was skewed towards topped seedlings, and it resulted in a slightly lower sensitivity. We end up with a few false positives as well, usually with seedlings that have more than one leader. However, overall, the results were excellent, and most of the errors could be corrected with additional training data. The results from the segmentation algorithm were equally impressive. This is the mask RCNN algorithm that looks to both detect seedlings but also to segment out the individual crowns. The test set accuracy in this example was 91%. We set our overlap threshold quite high because we want to do phenotyping, so only predictions that overlap ground truth by 50% are counted. But overall, we still see here very accurate segmentation of the crown area. Once again, small training data set, but most of the seedlings are identified with high confidence, and the key crown area that we're interested in for phenotyping was accurately segmented out by the mask RCNN algorithm. Aside from the loss of the height trial, the overall conclusions from the study were very positive. We demonstrated 2 new methods for imaging long seedling beds using direct georeferencing and panorama generation. Importantly, both could achieve success using a single flight of the UAV with just 40% image overlap. The PT GUI solution offered a low-cost, no-code solution to generating high-quality panoramas of the seedling beds. The darknet algorithm achieved 96% precision and was extremely scalable. In combination, both of these methods are likely to be able to image long seedling beds and count tens of thousands of seedlings. From a phenotyping perspective, the results were particularly encouraging. We validated microplot extraction in beds of radiata seedlings, and what this will let us do is set out large randomized trials in our open-grown nursery conditions, and it paves the way for automated analysis pipelines. The instance segmentation algorithms will further help this, and they can clearly delineate and detect seedlings at multiple ages. And I believe that the accuracy of both of these models would be further improved with additional training data. The key message I would like you to take away from today's presentation is that the tools and groundwork are ready for large-scale nursery phenotyping. We've met all the critical requirements for automation and phenotyping, and now what we need is a clear implementation pathway and a use case. And I hope to continue this line of research in partnership with Scion and the forest industry. Thank you for your attention today, and I look forward to taking your questions at the end of the session.